just in case you've never heard of me or Apical or whatnot, we're based out of Canby, Oregon. So just to give you the who, what, when, where, why of what Apical is and what we do. Anyway, we were established in 2017 as a lab that's analyzing plant fluids. Anyway, the leaf extract analysis that we have developed and are commercializing at this point is a horticultural analysis technique that analyzes primarily mineral content in the plant fluids. A lot of people would say, well, why do you want to do this? And so on and so on. But directly off of the frequently asked questions on our website, by using the mineral data, you can inform nutrient management decisions. It's the simplest way I can put it. And then by sampling and analyzing different parts of the plant, other agronomic principles can be utilized to determine nutrient flows, deficiencies, toxicities, tie-ups, imbalances, etc. Anyway, this topic may be somewhat a review for some of you folks, but I do think that there's going to be some information in here that maybe you haven't heard of before or maybe had not thought of when looking at a mineral report. So anyway, if any of you guys have further questions about Apical, I'd encourage you to either check out our website, which is www.apical-ag.com. I think it was actually on the first screen down there at the bottom. If you've never been to our website before, I'd encourage you to do so. There's a lot of information on there just on our methods and what we're doing and how to get started, how to get plugged in, and so on. And backing up a quick second, the first picture on this home slide here, picture sometimes speaks 1,000 words. And what you have here in this picture is two leaves that have been picked on the same day in two different parts of the field. One is in a treated area and the other is in a non-treated area. And basically what you're looking at is the difference in when we apply proper nutritional approaches to our crops, what can happen in the field. And as you can see from the first picture here, this is a blueberry leaf taken last summer, and you can see through one application after responding to mineral needs assessment that we had tested in the lab, you can see some significant growth and expansion of the leaf in this particular instance. So that basically sums up a lot of what the capabilities can do in a single picture. And it's important because, as many of you folks probably are aware, greater leaf area includes greater sugar production, greater photosynthesis, greater mineral transport, so on and so on and so on. Now, we're not just after gaining larger leaves, we're actually always after crop quality. But in a general perspective, by dealing with some of the issues that you might see every day out in your fields, you can achieve a much healthier crop, you can achieve larger leaf sets, you can achieve a number of different outcomes, too many to really list in a single slide. So anyway, moving on, I just wanted to share that picture and just kind of preface it, because this is something that we've been able to replicate over and over and over on a number of different crops in diverse soils and so on, utilizing the lab and the database reporting technology and so on. It allows you to make some visible differences in your crop, which I know is what a lot of folks are after. So anyway, so there's the who, what, when, where, and why, and we're based in Oregon, and so we're a younger lab. We've only been in operation. This is actually our third season, and every year we make new improvements and refine our technology, refine our methods, refine our reporting and data analytics. It's an ongoing project. We're really just trying to improve crop quality for growers and do the best job that we can to help growers improve their crops in a number of ways. So, okay, so why analyze the plant fluids? So as Steve was mentioning briefly, there's a number of different reasons why one person or another may want to analyze the plant fluids. The nutritional imbalances, deficiencies, toxicities, tie-ups, hidden hunger, evaluate fertilizer efficiency and delivery, studying interactions among nutrients, and so on. These are all general method and methodology reasons that folks would engage in a tissue analysis or a sap analysis or a leaf extract analysis or NIR type imaging, et cetera, et cetera. Fluids are constantly transported within the plant, and primarily they start in the root and they end in the fruit. And by studying that data down to as low of parts per million values as we can get, or sometimes parts per billion, we can actually begin to unlock some of the language that the plant is actually communicating with the soil in. And we call this plant-soil symbiosis, and it's a common opportunity and also challenge for a lot of growers to understand what's being translocated through the plant from the soil into the fruit. And without any sort of guide or map or any real relevant understanding, there's a lot of different outcomes. And one story I've told before is, and it's a common thing out there, you have a crop failure. I've had these things myself as a farmer in the past, and also as a manager. And oftentimes they are due to some piece of information that we just do not know how to access. We don't know where the deficiency came from. We don't know what started the disease. We don't know what brought in the insect. We don't know any of the drivers behind some of the challenges in our field often. So by getting down to brass tacks of actually what's going on in the plant leaves and what the fluids are actually being transported, or what is in the fluids that are actually being transported within the plant, we can gain maybe a leg up in terms of our production. So why do we choose to do things the way we do? We've found that by analyzing the fluidic data, we can develop nutrient management feeding programs that are accurate down to 1 ounce per fertilizer, and translate that into parts per million values in a particular crop. So this takes a little bit of practice, and it takes a lot of checking and rechecking and evaluation of different fertilizer programs, or not just fertilizer programs, just different approaches, soil types, etc. So anyway, there's a lot of variables going on, and so it's tough to sort through all that, and that's what we're attempting to do, is wade through these challenges. So anyway, but in short, the question that we always get is, why do my leaves look like this? Whether it's good on the left or really bad on the right, we see tremendous variation in crop leaves. Now, a lot of this, maybe some of you folks are aware of using, near infrared technologies or some of the other hyperspectral type technologies. A lot of those have very, very relevant information. But at the end of the day, what's causing these changes in the leaves is the mineral content in the plant fluids. And so all these photos here were actually taken a couple of weeks ago when I was out visiting a grower of mine that I work with in a raspberry block. And this raspberry block has been challenged significantly from a number of ways, but we've actually managed to, I would say, restore a fair amount of the genetic functional capacity of this crop over the course of the last 18 months. Now, that said, there's still field variations, and this is a constant challenge in a lot of different crops and a lot of different soil types, specifically those folks that have sandy soils, low organic matter. Field variations are a very, very real phenomena. So anyway, what we have then is all of these pictures were taken on the same day in the same block, and all of these are due to nutrient management variations in soil type. Nutrient variations due to soil type. All of these leaves are actually on the same nutrient program, believe it or not. So anyway, we have all these tools out there, but how do we actually implement some of them in order to get our leaves to look like the pictures on the left as opposed to the pictures on the right? And so this is what we've set out to do, and we like to think that we're actually making some significant progress. If you guys are out there and you're into crop management or nutrient management or agronomy or are agronomists or are intensive growers, you've probably seen these types of charts. And you look at the chart on the right and you say, well, gosh, I maybe have seen one or two or three of these things out in the field, but then maybe I try to correct it and maybe it doesn't work or what have you. And then you look at the picture on the left, and gosh, it's hard to make sense of all that information there. So there's a lot of really good information in a good Mulder's chart, but at the end of the day, you kind of go around in a wheel, and going around in a circle is not where you want to be in a profession where you maybe get one chance per year to make a crop improvement. So all day long, folks will send me or folks that I know, hey, what do you see here? Why are my leaves look like this? How do we change this leaf and make it look green again? How do we restore the crop potential? What's happening here? All these questions can be assisted by the technology that we're working with analyzing the plant fluids. Okay, so historically, a lot of folks have used tissue analysis in the past, and tissue analysis is great. And our leaf extract analysis is not intended to replace tissue analysis. Now, tissue analysis has a significant data set, a lot of scientific validation behind it. It's been in existence for over 40 years, and folks have used it to make crop improvements over the long term. The methodology in tissue analysis, the leaves are sampled and dried via microwave digestion. After that, the dry leaves are crushed and extracted with solvents, and the solvents are then analyzed on lab equipment. So it's a lab-based process that analyzes the dry matter. So at the end of the day, all of the leaves that you're looking at there are very dry and are getting ready to be then later on extracted in a tissue lab. So the reason why this is important, and I want to make some of these distinctions, is because we do get a significant amount of questions is, well, what's the difference between leaf extract analysis, sap analysis, tissue analysis? There's folks don't really necessarily understand a lot of this, and so I just want to go through this quickly to help you guys understand a little bit more about why we do what we do and how it differs. So sap analysis has been in circulation in the scientific community and being studied in various formats, I would say, since about the 90s. And a lot of you folks maybe have tried some of these techniques in the past. Brix meters, garlic presses, Hariba meters. These are really cool tools. And for folks that are out in the field and have, that want to be testing Brix, maybe some of you guys are in the wine industry that are already doing this. It's a really good tool to have as a Brix meter and a garlic press. You can always check, hey, is my Brix improving from 1 week to the next? Or is it improving from one season to the next? Or, gosh, why does the Brix of one cultivar look different than another? Or et cetera, et cetera. It can be a really useful approach to help getting started. Anyway, that said, there are some limitations within your typical sap analysis that growers use at home. And a lot of those are limited analytes, maybe not the most accurate type of equipment. Maybe, you don't have time to go out and, test 50 different varieties of 9 different crops and run those through your garlic press and so on, and maybe you don't have the crew to do it. So anyway, it is a really useful tool, and I would encourage everybody that is interested in crop management to get yourself a Brix meter. It's a very useful tool to understand, is my crop improving from a holistic perspective. So that said, like I mentioned, there's some limitations. And so in order to do this, we started looking at how do we access the depths of inside the cells of the plant. And so just comparing and contrasting briefly some of the, quote unquote SAP methods versus what we do in our lab is you look at the picture on the right here on this slide and you see that's what a lot of other folks do when they refer to SAP. They're crushing the leaves, they're pulverizing them, and then they're utilizing that juice. We take a slightly different approach, and if you look at these three photos and the inserts that we've put in the photos, these are actual leaves that were sent into our lab, and these are the actual physical samples that were extracted from those. So what you're seeing is the before and after photo of, I believe those are nectarine leaves, blueberry leaves, and hop leaves, and the actual fluid that came out of those leaves. And if you look, there's I don't know if you can read this on your screen, but there's before and after photos of each one of those. And believe it or not, those leaves on the right-hand side of each one of the photos have had the fluid pulled out of them and collected in the test tubes that are in the inset of the picture. Now, I make this distinction because the pure cellular fluid has a lot more accuracy in terms of being slightly different than your tissue calibrations. And the fluid is actually extremely concentrated because there's no vascular bundles, lignin, cellular wall structures, other parts of the plant organs that may skew the results and make them a little bit less pure or maybe more dilute. So anyway, above these three photos, in the upper right-hand corner, you see our adjuvant ICP that we use to analyze the primary amount of the plant nutrients. And we do have some other pieces of lab equipment that we utilize to do the bricks and the pH and some of the other analytes on the iodine, chloride. But the majority of the analytes are analyzed on the ICP. So why do we use ICP? ICP is utilized by the EPA to analyze sub-part per billion for most analyte quantitation. So what does that mean? You're getting extremely precise readings from the data that you get from us. So not only are we extracting the fluid in as pure of a possible way as we can, but we are also analyzing it in as accurate of a way as really known to the scientific community. And I get a lot of questions as to, how did you come up with all this? How did you do this, that, the other thing? Well, at the end of the day, it's been a group effort over the course of about 6 years to get to this point. And we have been extremely fortunate to have a number of classically trained physicists, chemists, biochemists, toxicologists, and application scientists to develop the method from start to finish in a way that we feel is extremely accurate. So anyway, we get a lot of questions on the, why the fluid actually out of the cell walls and so on. And that is, like I mentioned, it's just more concentrated and it's a bit more accurate to distinguish that, the characteristics of that fluid as opposed to the totality of the plant leaf that's been crushed through a garlic press or a juicer. Okay, before moving on, I just want to clear up a little bit more of the loose terminology. There's a few vocabulary words, and I'd encourage any of you guys that are really into this sort of thing to, to pull out a botany book or buy one on Amazon or, buy a plant physiology or a biochemistry book. There's a lot of information that can be captured and digested if you have a little bit of context for it. So anyway, the cytoplasm is all the material within the cell of a plant, and it's enclosed by the cell membrane. The chloroplasts are organs that capture energy from sunlight, convert it, and store the energy in ATP and free oxygen from the water in the plant cells. Ribosomes are minute particles that consist of RNA and associated proteins and are found in the cytoplasm. They essentially do perform RNA replication and transfer in order to synthesize all the proteins and so on that the plant needs in order to continue to grow. Guard cells are cells that surround the stomata of a plant that regulate the opening and closing of the stomata and facilitate gas exchange. So I wanted to just brief you guys on these terms because in the next couple of slides you're going to see some photos here. So continuing on as to what we're actually analyzing, why we're analyzing it, and so on. If you see this, the picture on the left where you see the cell wall of the plant, these are actual pictures of the plant. You see the cell wall, you see the fluid actually inside the cell and the chloroplasts that are associated with it. Now, you have a tremendous amount of biological plant activity within those cells, and that's what a plant is actually using to create fruit or to create the fluid that then later on becomes the fruit of the crop. On the left-hand bottom corner, you see a guard cell in magnification, and then just next to it, you see a inset diagram of some of the. It points out a couple of the actual organs of the guard cell. Now, I bring this up because there's often a lot of questions later on, and get into this shortly, about foliar feeding and how do we actually get foliar nutrition into the plant. And I know Dennis has done some great work on educating in previous webinars how to actually get nutrients through the guard cells and into the plant's vascular system. So off to the right-hand side, that's a further magnification of the cell wall in the middle, and then a further magnification of actually the ribosomes within the plant cell that are performing cellular replication. Okay, so just continuing on a little bit more, if you look to these diagrams on the left side, you see another sort of an infographic on what a guard cell is doing. Now, the guard cell is opening during the day and it's closing at night, and it's allowing gas exchange into the plant or moisture, as the case may be. Now, why is this important? Well, when you have mineral imbalances in the plant leaf and those guard cells open up, what happens? Disease gets inside there and it has a tendency to replicate, and it utilizes mineral imbalances within the plant cell walls as exploitive opportunities. And through those exploitive opportunities, then it begins to feed on the nutrients that are in the cell walls. So how do we assist the plant to fend off some of these problems? Well, a big part of that is by strengthening cell wall structures. So, you know, maybe other guys that are into agronomy have heard the importance of calcium, right? That's a very common theme. Well, calcium increases the cell wall strength. Hang on one second. Hang on, we've got someone calling in here. I apologize for the audio disruption. Anyway, back to the matter at hand here. So what you see is the cytoplasm in the middle photo here. The cytoplasm is what we're actually extracting and analyzing. So the liquid and all the DNA replication that's happening within the cell walls is actually what we are analyzing for mineral content. So with the mineral content, it has a tendency to as you're backing up a second, as your mineral content goes out of balance, the diseases, insects, et cetera, utilize the plant fluid as a food source. So how do we alleviate that? And this is what we refer to as plant-soil symbiosis. So the infographic on the right-hand side illustrates some of the theory behind why the plant, in the initial picture on the home screen, at the very first slide, how the plant can go from having a larger leaf to a smaller leaf and back. So when I refer to mass balance of nutrients, that's the total amount of nutrients that's actually available, or that's in the soil physically. Now, as many of you may know, due to soil microbiology, soil biochemistry, plant physiology, root health, so on and so on, there's a lot of limitations that may happen at different times of the year. So immediately, maybe you lose 20 to 30% of the functional capacity of your soil due to nuances within the soil's capabilities. Okay, so then the next thing that happens is, well, as some of those nutrient availability is limited, then you have a secondary limitation that happens, and that is due to the physiology of the plant. As you're doing this, you're continually limiting the capabilities of the plant without knowing it. So without having an optimal active microbiology that's at the proper carbon and nitrogen ratio, and continually spoon feeding nutrients to your plants in the form of decaying microbial metabolites and microbes themselves, you will have unknown or hidden hunger in your crop. And so if you look on the left, you have these basic soil influences that are always, always, always causing limitation on your crop unless you deal with them. So physical influences such as soil compaction, drainage, aeration, etc. Your biological influences, bacteria to fungi ratios, nutrient cycling, nematodes, the whole of the soil food web, protozoa, etc. And then obviously the mineral influences of the soil. Well, no one wants to grow 30% of their crop yield. And it's sad that a lot of crops around the world do. And this is why. And it is our goal to begin to restore these capabilities in a methodical way, utilizing the technologies that are available to us as scientists. The next piece to the puzzle is improper fertilization. So when you're talking about plant-soil symbiosis, there's nothing worse to plant-soil symbiosis than improper fertilization. And if any of you folks are certified crop advisors or agronomists or have gone to ag school, you've probably heard of the 4 R's: right source, right rate, right time, and the right place. Well, oftentimes we just don't know. And maybe we think we're doing the right thing and trying to implement the 4 R's, but we actually implement the 4 W's. The wrong source, at the wrong rate, in the wrong time, in the wrong place. And by understanding a deeper look at how the plants are translocating nutrients, as we'll see in a few minutes, we can begin to hopefully change the 4 W's back to the 4 R's. So I would contend that most growers have 1 or 2 problems with the issues of of improper fertilization. So here's a really common example that we see in low pH soils, as we have plants that have excess aluminum associated with them in the soil, or excess aluminum availability due to low pHs. We see the aluminum begin to limit the availability in the plant of phosphorus, boron, molybdenum, iron, calcium, and I left the rest blank because there's even more cascading deficiencies that can come from that. Why is this important? Well, as many of you know, phosphorus is crucial to ATP, and calcium is crucial to cell wall growth, and boron is crucial to sugar production. So as you limit all of those within the plant, you then begin to limit your root penetration strength. By limiting your root penetration strengths, you then make the plant more susceptible to root pathogens. So aluminum toxicity is something that we consistently see every week in, week out, in terms of our plant's capabilities to respond to challenges. And where they have aluminum, it's one of the biggest inhibitors of proper plant growth that we see on any lab test. The second major problem that we see in crops is excess sodium in the soil. So when you have excess sodium in the soil, the plant immediately tries to deal with soil salinity by increasing potassium uptake. And why is this important? Well, it increases, as many of you may know, potassium and calcium are directly antagonistic to one another. Because the calcium is decreased, you have increased leaching from the calcium. When you have decreased calcium uptake, you have decreased cell wall strength, decreased fruit wood, decreased bud count, and decreased crop yield. These are very, very real examples that we see every single day. Many of you may utilize a lot of copper fungicides, and I would contend that copper is a fantastic product when used in the right time, at the right amount, in the right way. When you have too much copper in the plant, you will limit iron and manganese uptake. You will reduce the splitting of the OH and H within the cells of the plant. You'll increase the pH in the plant cytostolic fluid, and you will decrease water holding capacity in the plant leaves and stems. All of this makes plants more susceptible to enophytic pathogens. So as the mineral content decreases or increases in an imbalance, decreases in total concentration or increases in imbalance, you have a longer term disease susceptibility that is developed within your plant. And the last one of the main toxicities that we deal with often is excess chloride. And a lot of folks say, well, chloride, do you actually have a correct reading there? Because they see very high numbers in their plants. And I would contend that our chloride analysis is extremely accurate and actually follows EPA guidelines and has been validated a number of times. So what we see in the plant is when you have excess chloride, it will immediately tie up calcium within the plant. It will stimulate sulfur and antagonize phosphorus, and which all of these do increases the need for increases the need for nitrogen, decreases the photosynthesis speed, decreases leaf size, and increases fruit quality, reduces fruit quality. So all of these toxicities are acting in a cascade of nutrient de-synergy. And so this is what's happening in your molder's wheel without you really fully grasping it potentially. And we didn't fully grasp it for years. And I would contend that it took 5 years to even identify the remote pathway that we wanted to take to get our lab off the ground. So how do we restore plant capabilities with mineral nutrition? Looking at the plant on the right, this is a picture that's a few years old of what happens when you hit the bullseye with crop nutrition. And the crop nutrition in this particular case is this was actually a combination of calcium and phosphorus that was applied to the root zone of a lemon tree. And this lemon tree then began to kick off leaves that were, 500% larger than the initial leaf set that the plant was putting out. Now, if that's not showing a reversal of crop issues and restoring genetic potential, then I don't really know what is. So now, in terms of crop yield, the genetic potential, I think we don't really have any understanding of, and it would be really good to track a lot of these over the long term because there's significant variations between varieties and cultivars and so on. But back to the topic here, by identifying the toxins in the soil and the plant and performing corrective measures to those, one can begin to restore this functional capacity. Now, the nutritional mineral balance is then achieved by performing the corrective measures. So by limiting your toxins and by addressing your deficiencies, whether that's done in sequence or at the same time, that can actually begin to unlock some of the challenges based on your crop. So as a review, and this information is all available on our website, and for those of you that were late in joining us, I'd again encourage you to go to the website at www.apical-ag.com. You've got your mobile nutrients that live in the phloem, and these are nitrogen, phosphorus, potassium, magnesium, chloride, iodine, sodium, and molybdenum. The deficiencies of the mobile nutrients are going to be indicated by lower ppm readings on the old leaves compared to the new leaves. And this is due to sluggish transport of the nutrients to the new growth. Now, what is causing that sluggish transport? Well, it could be indicated by a nutrient tie-up via your molder wheel. It could be an issue with the soil. It could be some sort of endophytic pathogen. It could be a range of reasons. A surplus of mobile nutrients are indicated by higher ppm readings in the old leaves compared to the new leaves due to lack of complementary nutrient synergy. Now, as some of you students of agronomy and ag may have already understood, there's synergy between elements such as calcium and silicon, or manganese and potassium, or iron and phosphorus, or zinc and phosphorus, or a whole range of other ones. Now, chemically, though, there's even deeper synergies that maybe we're not aware of, such as the synergy of chloride binding to calcium, or of sodium to driving potassium, or some of these other ones, such as aluminum and aluminum limiting silicon, etc. So when you have a surplus of mobile nutrients, that means that you don't need to feed any more of those nutrients at that given time, and that any crop problems that you're experiencing are due to some other reason. Looking at the immobile nutrients, calcium, silicon, sulfur, iron, manganese, zinc, boron, and copper, these deficiencies are indicated by lower ppm readings in the new leaves compared to the old leaves, as the plant is unable to transport them with new shoots. The surplus of immobile nutrients is indicated by higher readings in the new leaves compared to the old leaves.